All right, this is chapter 6 on bones and skeletal tissue. So we're going to learn about classification of bones and the parts of a bone and all that sort of stuff. And then the identification of bones in the human body will happen in the lab. So as always, here's our learning objectives. So let's talk about the functions of bone first. There are several important functions that bones provide for us. First of all, they support the body. If you didn't have bones, you would be a jellyfish. Bones facilitate movement. They work with the muscles and the tendons and the ligaments. Muscles are going to, or at least the tendon to that muscle is going to span a joint. The joints are where everything moves. And when that muscle contracts, it's going to cause movement of those at the joint of those bones. It helps to protect our internal organs, like the rib cage, our brain. It produces blood cells in the process of hematopoiesis, and it stores and releases minerals, in particular calcium, as well as fat. So when we look at the skeletal system, we have two big divisions that we look at. So the axial portion of the skeleton is in brown or tan, and the appendicular is in green when you're looking at the picture. So there are 206 bones in the adult human skeleton. Babies have a few more. Those bones fuse after they're born, right before they start walking, all that sort of thing. So they will eventually have only 206 bones as well. So back to axial and appendicular skeleton. Axial is everything in the middle. So the skull, the vertebral column, the rib cage, all that sort of stuff. And then when we talk about the appendicular, it's everything in green on the picture, and that's going to be your arms and legs and associated girdles. So a girdle, just like a girdle holds everything in, a girdle is what holds your arm. The pelvic or the pectoral girdle is what holds your arm in place, and that's going to be the collarbone and the scapula, clavicle and scapula. And then your pelvic girdle, which is going to be your hip, two hip bones that are going to hold your leg into place. So when we look at bones, we can classify them by their shape. And so there's 5 main shapes that we look at. So you're going to want to make sure you know the name of the shape as well as a description of it, what it does, and then some examples from it. So we'll start with a long bone. And so a long bone is longer than it is wide. Longer than it is wide. So these are all about leverage. These are the ones that we're going to have muscles attached between two of them spanning a joint. That's going to cause movement. So things like your femur and your tibia and your fibula. But bones that are not long also include things like your phalanges, your finger bones. Because remember what the definition is. The definition is longer than it is wide. It doesn't have to be necessarily really long. It just has to be longer than it is wide. So your finger bones are examples of long bones. Short bones really should be called square bones because they are cube shaped. And so they are approximately equal in their length and their width and their thickness. These are more about stability and support. They do allow a little bit of motion. So classic short bones are your carpals, which are your wrist bones, and your tarsals, which are your ankle bones. Flat bones are thin and curved. These serve as a point of attachment for muscles. They also serve to protect internal organs. So things like your sternum and your ribs and your scapula, your cranial bones, those are all examples of flat bones. Irregular bones are bones that didn't fit in any other category that we've already talked about. They have a complex shape. Typically, they are for protecting internal organs. So the vertebral bones and the facial bones are good examples of irregular bones. And then finally, a special type of short bone. Some books put sesamoid bones as a subtype of short bones, and some books have it separated out. Either way, it doesn't really matter. A sesamoid bone. A sesamoid bone is a small round bone that is embedded in a tendon. And so the big example in the human body of the 206 that we talk about are the patella. And so these protect the tendon from compression forces. However, for those of you going into rad tech, what you'll find out is that people build sesamoid bones in tendons that they put a lot of stress on. So commonly you'll see sesamoid bones in the elbow area for like bodybuilders because they've been lifting a lot of weight that their body actually builds a piece of bone in there because that's stronger than the tendon to help support all of the stress that's being put on that particular joint. Ice skaters, often you'll see them, and gymnasts often too, you'll see them in the ankles. So any place where people continuously put a lot of stress, not just one time or two times, but continuously, usually because of athletics, you'll see sesamoid bones. We're going to look at the anatomy of a long bone. So remember, these are longer than they are wide, and we are looking at a left femur here. So there's three components to all of our long bones. There is the diaphysis, which is the long cylindrical portion of the bone, the epiphyses, which are the top and bottom, the heads of the bone, and the membrane, which is the periosteum around the outside. So all bones have those features, and then they have some other features we're going to talk about too. So the diaphysis, as I mentioned, is the tubular shaft that runs between the proximal and the distal ends of the bone. So those two epiphyses that I just mentioned, one is proximal, one is distal. And again, it goes back to that idea to the point of attachment. So in this particular case, the one at the top is proximal because that is the head of the bone that's going to fit into the acetabulum of the hip bone. And then at the other end is the part that's going to articulate with the tibia and the fibula. So that would be the distal epiphyses. The hollow part in the very center of the bone, in the very center of the diaphysis, is called the medullary cavity, and it is filled with yellow bone marrow, which is just a real fancy thing called fat, right? So you got fat inside your bones. The wall of the diaphysis is composed of dense and hard compact bone. I've talked about the epiphyses already. This is the flared or wider section at the end, each end of the bone, and it's filled with a different kind of bone called spongy bone, and we'll be talking about that in just a few minutes. In that area, we don't have yellow marrow, we have red marrow that fills that area. These are covered with articular cartilage on the ends, which is hyaline cartilage. It forms the joint surfaces and helps to reduce friction. That articular cartilage that is on the cap of the bone, if you look all the way up at the top and down all the way at the bottom, it's kind of that like sky blue color. That's the remnant of the fetal skeleton. So the fetal skeleton actually started out as a hyaline cartilage model or precursor and slowly converted over to bone. And what's left is those two pieces. So let's look at this epiphyses a little bit closer. So each one is going to meet the diaphysis at the metaphysis, the narrow area that contains the epiphyseal plate. That's the thing that's most important to us. So I want you to look for that clear area that they show on here, and they're pointing it out, is called the epiphyseal line in an adult. In a growing child, it's called the epiphyseal plate or the growth plate. So remember I said you started out as a hyaline cartilage mini-you, and it's going to convert over to bone, and where that conversion is happening is at that epiphyseal plate. Once you have reached the end of puberty and your bones are no longer getting any longer, that plate is going to seal and become the epiphyseal line. And that's what it's telling you out here. When the bone stops growing in early adulthood, which for young ladies is usually about the age of 21. For young men, it's usually, or young ladies, it's about the age of 18. For young men, it's usually about the age of 21, but there's some variation in that. The cartilage is going to be replaced with osseous tissue, and that plate becomes the epiphyseal line. The membranes that are associated with a long bone. So the medullary cavity, the hollow part that's in the middle, it has a lining that's very delicate that's called the endosteum, endo meaning inside, ostia meaning bone. And this is where bone growth, repair, and remodeling is going to occur. So the inside of the bone in the medullary cavity has a lining. And then the outer surface of the bone is covered with a fibrous membrane connective tissue structure that's called the periosteum, peri meaning around. The periosteum is going to contain blood vessels and nerves and lymphatic vessels that are going to nourish the compact bone. The tendons and ligaments are also attached to the bone at the periosteum. So I kind of think of it as the bone covering, if you will. And they've got it peeled back on the picture so you can see that. So that's what a long bone looks like. Let's look at a flat bone for just a minute. So these are ones, for instance, like in your skull. So they've taken a little piece of one out to show you what it looks like. And it's made different. It looks like a bone sandwich. So you have a layer of spongy bone in the middle. They call that the diplo. And on either side of that, you have a hard, smooth bone called compact bone. And in between there, you have red bone marrow. So now let's move on to what makes the skeletal system so fun, and that's bone markings. So bone markings are all the lumps and bumps and grooves and holes that you find in a bone, and they are consistent across individuals. And so those of you that are going into rad tech, this is like super, super important for you. This is all about you. Those bone markings are how you make identifications, because those things are the same. Other things can be different, but you look for those bone markings, and there are a bunch of them, and we're going to learn some of them in this course. There are three general classes of bone markings. There are articulations. Articulations are where two bones are going to join together, right? So articulus in Latin means joint. There are projections, and this is a piece of bone that projects above the surface or out from the surface of the bone. And these are typically attachment points for tendons and ligaments. And then there's holes. These are openings or grooves in the bone that allow blood vessels to enter. So bone is alive. It's not this dead structure inside of you. It is alive. And so it has a blood supply and a nerve supply. If you've broken a bone, you know that because it's very painful. So here are some examples of the names of the markings and the description of what they are, and just an example. There are dozens of examples of these. But let's take a look at some of these because you're going to want to learn some of these. I always in lab, you basically learn these as you learn the markings because you're looking at a condyle, for instance, and you're like, oh, this soft, this rounded surface that I see right here. And you're looking at the bottom of the skull and you think to yourself, ah, I totally get what this is. This is a rounded thing. So you kind of learn them as you're learning the names of the markings, but it does help to look through these and make sure that you have a general understanding what each of these mean. So that is your little project for yourself to do, is to go through here. Flashcards are really, really good for this. So you would want to know what the general name of that group of markings is and what they are, be able to describe them, and then be able to give some examples. But you're going to see plenty of examples in the lab next week. The chemical composition of bone. So bone is made of both organic and inorganic compounds. The organic compounds include the cells and the protein fibers. So bone contains a relatively small number of cells in relation to, in comparison to other tissues that we've looked at. Those bones are actually trapped in a matrix of collagen fibers that provides the surface for the inorganic salt crystals to stick to. So the two of them are meshed together. The inorganic compounds include the hydroxyapatites. These form when calcium phosphate and calcium carbonate combine. So hydroxyapatites incorporate other organic salts like magnesium hydroxide, fluoride, sulfate. These all are things that crystallize or calcify onto their collagen fibers. The hydroxyapatite crystals give bones their hardness and their strength, and the collagen gives it some tensile strength and makes it stress resistant. So bones have a little bit of bend to them because of collagen. Not a lot of bend, but a little bit. If they didn't, the first time you were going for a jog and you came down hard on your foot, you would shatter all the bones in your legs. So collagen is what gives that bone a little bit of give. The different types of cells that we would expect to find in bone tissue. So there's 4 types of cells that we expect to find, and each of them has its own basic function. The osteogenic cells are going to be the ones that are stem cells. The osteoblasts are going to be the ones that build new bone. The osteocytes are our mature bone cells, and osteoclasts are going to be the ones that break down or resorb bone. So let's look at our osteogenic cells first. These are undifferentiated stem cells, and they have a very high mitotic rate. So these can become any type of cell because they are a stem cell. So they can become any kind of bone cell except for one. This is the only bone cell that actually divides. So the other cells that you see are all derived from these particular osteogenic cells. These immature osteogenic cells are found in the deep layers of the periosteum and in the marrow, just basically sitting there waiting until they need to be available to become a mature cell. They're going to differentiate and and divide into an osteoblast. Osteoblasts, these are the cells that are responsible for building new bones. So osteoblasts build. Osteoblasts build. They are found in the growing portion of the bone. This includes the periosteum and the endosteum. These guys don't divide, but they synthesize and secrete the collagen matrix and the calcium salt. And remember, osteoblasts build bone. It's hard to say all those B's that are in there. Osteocytes are the mature bone cells. This is the most common type of bone cell that you'll see. Kind of looks like he's running there, doesn't it? Each osteocyte is located in a space called a lacunae, just like we saw with the cartilage, and is surrounded by bone tissue. So osteocytes, their function is to maintain the mineral concentration of the matrix and they do that via the secretion of enzymes. They communicate with each other. So if you think about it, osteocytes are they originally were osteoblasts, and they started to secrete the matrix, and they basically built themselves into their own little house. And that little house is called the lacunae, and then they're surrounded by matrix. So they're not in contact with one another the same way a skin cell is in contact with the skin cell next to it. But they're able to communicate with each other through these little hair-like openings, channels in the bone. They have long cytoplasmic extensions that you can see there, and then they extend those through the canaliculi, which are these little little openings, little tunnels, teeny tiny tunnels running between osteocyte lacunae, and they're able to communicate with each other. They communicate via chemicals. And then finally, osteoclasts. These are responsible for bone resorption or breaking down. And so they are continually breaking down old bone while the osteoblasts are continually making new bone. And as those osteoblasts make new bone, they build themselves into that and become an osteocyte. So there's a balance all the time between the building up by the osteoblast and the breaking down by the osteoclast. It has to be constant. If one gets out of balance, one's building more or one's breaking down more, you can imagine there's all kinds of diseases associated with that. Osteoporosis is a classic example. This constant reshaping of bone is called remodeling, and bones are always doing this. They're always breaking down and building new bones. So some bones take longer to do this, like a femur. It might take 7 years to completely remodel that whole bone. Other really small bones, like a little wrist bone or something, might only take a year. Your bones are constantly doing this, and that's what keeps them strong and healthy. So there's those cells again. Osteogenic cells, those are our stem cells. So you have the cell type in the first column and their function and where we could expect to find them. There's two different types of bone. There's compact bone, which is on the outside of our bones. And this is like a smooth layer that's on the outside of the bone. It is made of microscopic structural units called an osteon or a Haversian system. Osteon's a lot easier to spell, so go with osteon. All right. So I want you to kind of imagine it like this. Let's think about a long bone like your femur, right? So the whole outside of it is these long columns, these cylindrical columns that are all scrunched together. It's almost like if you think about a whole package of spaghetti, right? Uncooked spaghetti. Each one of those individual spaghettis would be an osteon, right? And so you have a whole bunch of those surrounding the medullary cavity. So they're running the length of the bone. So the osteon, when we look at it in cross section, is composed of concentric rings of calcified matrix that are called the lamella. So when we look at one of these rings, so you can pick any one of these rings, there's one that's got a circle around it. So when you can look at that circled one, running right up the very middle is a hole, and that hole is called an osteon. is called the central canal, sorry, of the osteon, and that's where the blood and the nerve is. You can see the yellow nerve and then the artery and vein that are in there. And then you notice that there's rings going around that central canal, those concentric lamella that are going around and around and around. That whole thing would make one osteon. So again, the compact bone is running the length of each osteon, and you have the central canal, also known as a Haversian canal. Again, central canal is a lot easier to spell. Haversian, if you choose to get fancy and use that name, that is someone's name, so you would need to capitalize that if you're going to use that term. Again, blood vessels, lymphatics, nerves, all that sort of stuff are found in there. The blood vessels and nerves are going to branch off of that central canal at right angles, and they're going to go through the perforating canal. The alternative name for perforating canal is Volkmann's canal. Again, that is somebody's name. If you want to use that, make sure you capitalize it. And those are going to extend out to the periosteum and the other direction to the endosteum. So you can see endosteum. So you can see that when you look down near the bottom of this picture, you can see the central canals going up and down, and then the crossbar in between them, that's going to be your perforating canals or Volkmann's canals. So a close-up of our osteocytes, what they look like. So we're looking at a cross-section of an osteon so that we can see what's happening with our cells and everything. And in the very middle, let me draw your attention to the central canal that's in the very middle there. And then you see all of those purple structures. All of those purple structures are an osteocyte, a mature bone cell, sitting in its lacunae, its little jacuzzi area that's around it. And you see all of those, what look like spider legs going everywhere. Those are the canaliculi. Those are the little communication tunnels. They're like hair thin, right? They're really, really thin. And again, that's how nutrients are going to be transported, and it's also going to allow for the cell to communicate with other cells, because cells like to talk to each other, and any waste that the cell is making, because all cells make waste, are removed that way as well. So spongy bone, we saw some spaces where spongy bone was, and it's called that because it looks like a sponge. The whole name for this was cancellous, I believe. Spongy is way easier to pronounce and to spell. So spongy bone has osteocytes in it as well, and they're in lacunae, but they're not arranged in those concentric circles that we just saw in the compact bone. So the lacunae and the osteocytes are found more in a lattice-like network that literally looks like a sponge, like what you keep next to your sink if you use a sponge. It looks like that. And you've got the matrix-like struts and plates that are called the trabeculae. That trabeculae are going to form along lines of stress and provide strength to the bone. To me, what spongy bone looks like, there's a picture of it, the little box. What it looks like a sponge, but it also looks like, if you've ever looked on the edge of a corrugated box, right? You look on the edge and where you can see the insides. That's what it looks like. And all those going back and forth pieces of cardboard inside the two walls of cardboard, that's what gives that cardboard box strength, those struts and plates. And that's the same thing that spongy bone does. So spongy bone is going to be found in places where you have external forces coming from a lot of different directions. So look at the example there. Again, we're looking at a left femur. And so when we're looking at that femur, what we can see, actually we're looking at a right femur, sorry. We're looking at a right femur on that particular picture. What you can see is that area where the spongy bone is, forces coming from a lot of different directions, because that is where your leg bone articulates with your hip bone, and when you take a step, force is coming in from a lot of different directions. If that was just compact bone, that would probably not be strong enough, but because you've got this spongy bone in that area, and it can take force from a lot of different directions, it's actually a lot stronger. How are bones formed and how do they develop? So in the early stages of embryonic development, the embryo skeleton, as I mentioned, is fibrous membranes, which we'll talk about in a few minutes, and hyaline cartilage, which I talked about already. By the 6th or 7th week of embryonic development, the actual process of bone development, ossification, and osteogenesis begin. There's two different osteogenic pathways that we're going to talk about: intramembranous ossification and endochondral ossification. The mature form of the bone is the same regardless of the pathway that produced it. So intramembranous, let's look at the word first. Intra means within, membrane means a membrane, ossification means to convert into bone. So this is when we start with a sheet of fibrous connective tissue, that's the template, and the bone is going to be developed from that. So bone cells develop directly from the sheets of the mesenchymal or undifferentiated connective tissue. This is how the flat bones of the face and most of the cranial bones and the clavicles or your collarbones are going to be formed. All right, the process of intramembranous ossification is going to begin when mesenchymal cells in the embryonic skeleton are going to cluster together and start to differentiate into osteoblasts in a cluster that's called the ossification center. The osteoblasts are going to begin secreting osteoid, which is uncalcified matrix. That's going to calcify or harden as mineral salts are deposited onto it. And it's going to trap those osteoblasts within that matrix that it's making. Once they're entrapped, the osteoblasts are going to convert and become mature bone cells called osteocytes. The osteogenic cells in the surrounding connective tissue differentiate into new osteoblasts. The osteoid that's secreted around the capillaries is going to result in the trabecular matrix, while the osteoblasts on the surface of the spongy bone become the periosteum. The periosteum then creates a protective layer of compact bone superficial to the trabecular bone. The trabecular bone crowds near the blood vessels, which eventually condense into the red marrow. So that's how we get what they call a membrane bone, right? So bones of everything from your neck up is a membranous bone, with the exception of those clavicles, your collarbones, right? Those are below the neck, and they're going to be a membranous bone as well. So endochondral ossification. Now let's talk about that. Now let's look at this word first. Endo within, chondral means cartilage. Ossification, of course, means the process of making bone. So this is replacement tissue that's happening. All right, so we're replacing the cartilage mini-u with bone. Okay, this happens during fetal development. The framework's laid down that determines which bones are going to form. All right, let's talk about the steps of endochondral ossification. This is where we're replacing the hyaline cartilage precursor skeleton with a bone skeleton. So you start out with the little mini-me bone up there in the very top, and it's all made of hyaline cartilage. And so the cells that are in the very middle in the medullary area, they're going to start to die, and blood vessels are going to start to invade the area. And so you're going to have what's called the primary ossification center. That's the third little picture that you have there, or C. All right. And they're going to make spongy bone in that particular area because we're in the medullary cavity. That process is going to continue where we've got bone cells that are starting to build bone at that epiphyseal line or plate at this particular point. And as they build more bone, they push the head, each of the epiphyses, away from one another. So they keep turning over this cartilage and converting into bone. And they just keep doing that and doing that and doing that. And as they're doing that, the bone is just pushing the two ends further and further and further away. And so that you'll have that primary ossification center, which is in the center of the diaphyses, and then you'll have some secondary ossification centers that begin in the heads of the bone or the epiphyses in the same way. So that process is going to continue until we've converted that entire bone over into that cartilage model over into bone. So we've talked about how bones get longer, and that is what we're looking at here. But now let's look at it on the microscopic level. So the epiphyseal plate is this area where all of this growth of the long bone is happening. So once we have started this process, that process is going to continue until the end of puberty. So on the epiphyseal side of the epiphyseal plate, more cartilage is being formed. And on the diaphyseal side, that cartilage is being ossified. So we're just basically turning over this cartilage all of the time. And as we do that happen in each end, we add more to the middle and push the two ends further away from one another. So there's just a picture of showing you that idea of how the bone is just going to get longer and longer and longer. And this is all happening at the growth plate or the epiphyseal plate. So we talked about how bones get longer. Now we have to talk about how bones get thicker or wider. So there is a balance that's happening here between the osteoclast and the osteoblast activities. Remember, the osteoblasts are building bone and the osteoclasts are breaking the bone down. So osteoblasts are depositing new bone layers on the outside of the bone, and at the same time, osteoclasts are breaking down old bone layers on the inside of the medullary cavity so that the thickness of the wall of the bone stays the same, but the overall effect is for the bone to get wider. If the bone didn't take away from the inside while it was adding to the outside, it would just get too heavy and your muscles wouldn't be able to move that around. I mentioned the idea of bone remodeling earlier. This is the process where the matrix is broken down or resorbed on one surface of the bone and deposited at another. And so this constant process of remodeling, just like you remodel your house and you take out the old and you put in the new, and sometimes you use parts of the old in the new and you recycle some of it, the same sort of thing is happening with your bones. This primarily takes place during a bone's growth. However, in an adult, this is how remodeling happens to replace any old or damaged bone. This is what happens if you have an injury and you break a bone. Any of those kinds of things, the bone is going to have to be remodeled and reworked. This is what helps to maintain the homeostasis of our bones. Bones are really important. If your bones aren't healthy anymore, then it's going to make it really, really hard for you to do your normal activities. Nutrition in bone tissue. Vitamin D is important for calcium reabsorption in the small intestine. So you got to have vitamin D for that. In addition to vitamin D's role in calcium reabsorption, it also plays a role, though it's not clearly understood, in bone remodeling. So vitamin D is really important for keeping your bones healthy. Some of the hormones that are associated with bone tissue, the pituitary gland in your brain is going to secrete growth hormone, which, as its name is implying, is going to control the growth of bone several ways. Thyroxine is a hormone that's secreted by your thyroid gland, and that's going to promote osteoblastic activity and the synthesis of new bone matrix. During puberty, the sex hormones, estrogen in females, testosterone in males, is going to promote osteoblastic activity and produce more bone matrix, and is responsible for the growth spurts that we see during adolescence. What about some hormones that influence the osteoclast? This is the ones that are breaking bone down. So bone remodeling requires osteoclast to resorb. That word is always confusing because people want to say reabsorb, but it's resorb, which means to break down any unneeded damaged or old bone, and the osteoblasts are going to lay down new bones. There's two hormones that are classically responsible for doing this. This is the parathyroid hormone, PTH, and calcitonin. So let's talk about how we keep calcium in a homeostatic range. So just like you have to keep your blood glucose in a homeostatic range all the time, and that's accomplished through insulin and glucagon, we have the same thing going on with our calcium levels in our blood, and this is going to be accomplished through those two hormones, parathyroid hormone and calcitonin. So starting at homeostasis, which is the horizontal line in the middle there, let's say you have an increase in your calcium level. The thyroid gland is going to release calcitonin. Calcitonin puts calcium into storage, right? And it's going to put it into storage, and it's going to cause the osteoclast activity to decrease. when the osteoclasts are breaking down bone, they're releasing calcium, and we don't need any more calcium. We have plenty. So the osteoclasts are going to be inhibited. The calcium reabsorption in the kidneys is going to decrease. So we're going to release more calcium in our urine because we have plenty, and the calcium blood levels are going to go back down to where they're supposed to be, or back into the homeostatic range. What happens when we have a decrease in our calcium levels? In this case, a different set of glands, the parathyroid glands that are on the back of your thyroid gland, are going to release PTH, or parathyroid hormone. And that parathyroid hormone is going to cause the osteoclasts to start breaking down bone to release more calcium. It's going to cause more calcium to be absorbed from the urine at the kidneys, or by the kidneys in general. So uh, we aren't losing as much calcium in our urine, and it's going to cause us to uptake more calcium from our diet that is passing through our body. And that effect is to cause the calcium levels to increase. All right. So there's what we call the homeostatic mechanism for calcium control. Calcium is really important for bones, but we're going to find out it's super important for muscles and for nervous system as well in upcoming chapters. Some bone disorders to be familiar with. Hypocalcemia is when we have too low of a level of calcium, and that can cause bones to become brittle. And the causes of this can range from somebody's hormonal imbalances, like I was just talking about with calcitonin and parathyroid hormone, or their diet. So if you want to make sure that you don't have problems with hypocalcemia, then I highly recommend you eat more ice cream. Osteoporosis. I think most of us are familiar with osteoporosis as a disease. It's characterized by a decrease in bone mass that occurs when the rate of breaking the bone down exceeds the rate of making new bone. And this commonly occurs as we get older. Women start to lose bone mass more quickly than men because their skeleton is smaller typically. This actually starts in perimenopause in their mid to late 30s. But it really becomes obvious when they get into their 50s. That 50s is the approximate age that women go through menopause. Um, as long as a woman is not in menopause, she's still producing lots of estrogen, and estrogen helps to maintain her calcium levels. Once she stops producing estrogen, then she doesn't have that maintenance factor in there anymore, and so that's why you associate osteoporosis with menopause. The current treatment for this is to use certain drugs, biphosphonates, calcitonin, estrogen, those kinds of things, for women only. Too much estrogen for men can lead to prostate cancer. All right, so that's the end of this particular chapter. You'll want to make sure you study through all of this and take your quiz. See you for the next chapter.